So hello students, welcome to V Karnataka. ನಾನು ನಿಮ್ಮ ಮಾಸ್ಟರ್ ಟೀಚರ್ ಬಸವರಾಜ್ ಸರ್. ಸ್ಟೂಡೆಂಟ್ಸ್, ಈ ಸೆಷನ್ ಇದು MCQ ಸೆಷನ್. ಸೊ ನಮ್ಮ ಬಯಾಲಜಿಯಲ್ಲಿ ನಿಮ್ಮೆಲ್ಲರಿಗೂ ಗೊತ್ತು, ನಾನು ಗೋಪಿಕಾ ಇಬ್ಬರು ತಗೋತೀವಿ ಬಯಾಲಜಿನ, ರೈಟ್? ಸೊ ವಿ ಆಲ್ವೇಸ್ ಡಿವೈಡ್ ಅ ಚಾಪ್ಟರ್ಸ್. ಸೊ ಟುಡೇ ವಿಲ್ ಬಿ ಡೂಯಿಂಗ್ ದ MCQ questions from the chapter sexual reproduction of flowering plants, human reproduction and also principles of inheritance. Some of the questions are previous year question, andre exam al bandittu munche. Some of the questions are very new questions. So students, quick agi ellaru attendance na mark ಮಾಡಿ, that is video na like ಮಾಡಿ, ellaru comment ಅಲ್ಲಿ ತಿಳಿಸಿ ನನಗೆ if I am audible and visible to all of you. With that being said, students, video na share ಮಾಡಿ ಎಲ್ಲರಿಗೂ, because this is a new channel tane. This is a new channel. So in this channel we are trying to do your KCET as well as board preparation. PUC board agli illa KCET agli. So KCET will be doing it eventually, but right now the main focus is the boards. Nange ellarigu gottu nim board start agide agle ಕೆಲವು ಕಡೆ, but biology is still remaining, I believe. So these MCQs will help you to get those 1 mark question which is very easy to obtain. Okay, now students, quickly everyone in the chat, as soon as a question appears on your board, I want to see the answer. Done? Deala? Pakeli students ellaru deala? ಸೊ ಕ್ವೆಶನ್ ಬಂದ ತಕ್ಷಣ ಆನ್ಸರ್ ನೀವು ಹೇಳಬೇಕು ಪಟ್ ಪಟ್ ಅಂತ, ಸರಿನಾ? ಬನ್ನಿ ಸ್ಟಾರ್ಟ್ ಮಾಡೋದ ಫಸ್ಟ್ ಕ್ವೆಶನ್ ಇಂದ. ವಾಟ್ ಇಸ್ ದ ಫಸ್ಟ್ ಕ್ವೆಶನ್? ದ ಫಸ್ಟ್ ಕ್ವೆಶನ್ ಸೇಸ್ ಡ್ಯಾಶ್ ಆಫ್ ದ ಪೋಲನ್ ಗ್ರೇನ್ಸ್ ಡಿವೈಡ್ಸ್ ಟು ಫಾರ್ಮ್ ದ 2 ಮೇಲ್ ಗ್ಯಾಮೆಟ್ಸ್. ಸೊ ನಿಮ್ಮೆಲ್ಲರಿಗೂ ಗೊತ್ತು, ಇನ್ ಅ ಪೋಲನ್ ಗ್ರೇನ್ ದೇರ್ ಆರ್ 2 different cells. All of you are aware, right? In a pollen grain there are 2 different cells. One is vegetative cell, one is generative cell. Now the question is asking which of the two cells will divide, giving rise to 2 male gametes. Is it option number A, option number B, option number C or option number D? Quickly everyone in the chat. Students, ಈಗಲೇ ಅಂತ ಇದ್ದೀನಿ, ಮಾರ್ಕ್ಸ್ನ ಟ್ರ್ಯಾಕ್ ಮಾಡೋದ. I want to know at the end of the MCQ session how many questions you are able to solve. So if you are able to solve, +1 ಅಂತ ಎಲ್ಲರೂ ಚಾಟಲ್ಲಿ ಹಾಕಿ, ಬೇಗ ಎಲ್ಲರೂ +1 ಅಂತ ಎಲ್ಲರೂ ಚಾಟಲ್ಲಿ ಹಾಕಿ, +1 for every right answer. No negative marking. Chill. Sarna. So what is the answer? Students, all of you know the generative cell. All of you know the generative cell will divide into 2 cells eventually. It will divide into 2 male gametes. Divide into 2 male gametes. So what is the answer? Answer is option number B. So ಯಾರ ಕರೆಕ್ಟ್ ಆನ್ಸರ್ ಹೇಳಿದ್ರಿ? +1 in the chat everyone. ಎಲ್ಲರೂ +1 ಅಂತ ಹಾಕಿ. ಕರೆಕ್ಟ್ ಆನ್ಸರ್ ಮಾಡಿದರೆ ಮಾತ್ರ ಸ್ಕ್ಯಾಮ್ ಎಲ್ಲ ಮಾಡಬೇಡಿ, ಸರಿನಾ? ಸೊ +1 in the chat. Now next question. Intine of a pollen grain of, is it the inner layer of the pollen grain, thin and continuous layer, made up of continuous layer, or all of the above? 10 seconds, go. 10 seconds. Depending upon how this question happens, it can be 1 hour, but it can be 45 minutes, it can be 30 minutes, depending upon your rate of solving the questions. ನೀವು ಬೇಗ ಸಾಲ್ವ್ ಮಾಡಿದ್ರೆ ಕ್ವೆಶನ್ ಸೆಷನ್ ಬೇಗ ಮುಗಿಯುತ್ತೆ. ನೀವು ತುಂಬ ಟೈಮ್ ತೊಗೊಂಡು ಆರಾಮಾಗಿ ಮಾಡಿದ್ರೆ ಲೇಟ್ ಆಗುತ್ತೆ. ಸಿಂಪಲ್. ಓಕೆ, ಸೊ ಫಸ್ಟ್ ಆಫ್ ಆಲ್ ಸ್ಟೂಡೆಂಟ್ಸ್, all of you know a pollen grain has 2 different layers. All of you tell me in the chat, what is the outer layer called as? All of you tell me, if this is the pollen grain, the pollen grain is the outer layer which has ornamentation. The outer layer is called as what? The outer layer is called as exine. The inner layer is called as intine. Inner layer is called as intine. First one is correct. The thin and continuous layer, yes, it is thin and continuous layer. Okay, now students, you tell me, is the exine continuous? Simple question. Is the exine continuous? Obviously not. In the entire exine there are small holes in the exine. Can you find small holes in the exine? They are called as germ pore. Yes or no? So germ pores are present in the case of exine, but in the case of intine, nothing. Intine is continuous layer, so it is correct. Okay, it is made up of made up of cellulose and pectin. It is made up of cellulose and pectin. Correct or not? Tell me. It is made up of cellulose and pectin. Now you tell me students, what is exine made up of? Quickly in the chat. What is exile made up of? Exile is made up of a chemical substance. ಅದ್ರ ಮೇಲೆ ನೀವು ಆ್ಯಸಿಡ್ ಹಾಕಿ ಬೇಸ್ ಹಾಕಿ ಅದ್ರ ಮೇಲೆ ನೀರು ಹಾಕಿ ಎಲ್ಲ ಹಾಕಿ ಏನೂ ಆಗಲ್ಲ. That is called as sporopollen. So sporopollen is present in the case of exine, but the intine is made up of cellulose and pectin. So what is the answer? All of the above. So students, quickly marks alli chat allardu. What are your marks? +1 or +2? Quickly. So answer is all of the above. All of the above. Next question. The nutritive tissue of the anther is. So all of you know the anther wall. All of you know the typical anther wall, where we have the outer layer is what? The outer layer is called as epidermis. Then we have endothecium. Then we have middle layer, and finally the innermost layer is called as tapetum. The innermost layer is called as tapetum. Now out of this 1, 2, 3 and 4, they are asking you which layer provides nutrition. Which layer is responsible for pollen kit? Which is the layer? Students, ನಿಮ್ಮ NCERT ನೀವು ಓದಿದ್ರೆ ನಿಮ್ಗೆ ಗೊತ್ತಿರುತ್ತೆ. Out of this all 4 layers, the outer 3 layers are mainly for protection and dehiscence. Protection, while the innermost layer, the tapetum is for nutrition. Tapetum is for nutrition. So what is the answer, students? Answer is option number B. Okay. So tomorrow is physics exam, but I'm still here. Don't worry. One quick bio revision will happen. Then you go study physics. See, bio is ಒಂಥರ ಹೆಂಗಂದ್ರೆ bio ಒಂದು ರಿಫ್ರೆಶಿಂಗ್ ಆಗಿರುತ್ತೆ. ಇದು ಸ್ವಲ್ಪ ಜಾಸ್ತಿ ಥಿಂಕ್ ಮಾಡೋದು ಬೇಕಾಗಲ್ಲ. ಆನ್ಸರ್ ಸುಲಭವಾಗಿ ಬಂದ್ಬಿಡುತ್ತೆ. ಅದೇ ಫಿಸಿಕ್ಸಲ್ಲಿ ಸ್ವಲ್ಪ ತಲೆ ಕೆರ್ಕೋಬೇಕಾಗುತ್ತೆ. ಸೊ bio is very simple. Solve the questions, gain some confidence, then go back to doing physics. Okay. Next question, students. Now, a typical female gametophyte. A typical female gametophyte is. So all of you know the female gametophyte is the embryo sac. It is the embryo sac. Now in embryo sac, how many celled it is and how many nucleated it is? Okay. Now if you look at typical structure of an embryo sac, can I tell? Here we have 1, 2, 3. Okay. 3 cells, 4, 5, 6, 7. So can I tell it is 7 celled? 7 celled. But how many nucleated? 1, 2, 3, 4, 5, 6, 7, 8. There are 2 nuclei in the center. So can I call it 7 celled, 8 nucleated. 7 celled, 8 nucleated cell of structure of the embryo sac. 7 celled, 8 nucleated. Clear? Rohan, don't worry. Bio is very easy. You watch our lectures, you watch the most repeated topics in biology, watch the 5 marks question, watch the 2 mark and 3 mark question, and watch the MCQ questions. ಇದು ಎಲ್ಲ ಸೆಷನ್ಸ್ ನೀವು ನೋಡಿದ್ರೆ ಎಲ್ಲ ಸೇರಿಸಿದ್ರು ಕೂಡ exactly you will have 3 hours of bio. If you watch 3 hours of bio, I am very sure you will not fail in biology. Okay. Now, next question, students. The total number of nuclei involved in the double fertilization in angiosperms is. Number of nuclei involved in the entire double fertilization. Now students, you tell me, in the syngamy, how many nuclei are there? In syngamy, how many nuclei? We have one male nuclei plus 1 female nuclei, that is the egg. Okay, this will result in the case of zygote. Okay. Then in triple fusion we have 1 male nuclei again, 1 male nuclei plus 2 polar nuclei. These 2 polar nuclei are basically the central cell, which will give rise to the endosperm eventually. Now you tell me, how many total nuclei? Tell me, how many total nuclei is there? 1 nuclei here, second one here, 2, 3, 4, 5, 4 and 5. So in total, how many nuclei? 5 different nuclei are there fusing here. So what is the answer? Tell me, what is the answer now? Answer is option number D. That is 5 nuclei. 1, 2, 3, 4, 5. Okay. Next question. Okay. Now here I will tell you a small trick. Ready? One small trick is there. I know many of you would have faced difficulty in finding out what is radical, what is plumule, what is coleoptile, where you get coleorhiza. Okay. Listen to me very carefully. Coleoptile and coleorhiza are protective sheets covering dash and dash respectively. ನಿಮ್ಮ ಎಲ್ಲರಿಗೂ ಈ ಕನ್ಫ್ಯೂಷನ್ ಆಗೇ ಆಗುತ್ತೆ. ಬೆಟ್ಸಿ ಕೇಳ್ತೀನಿ, ಈ ಕನ್ಫ್ಯೂಷನ್ ಆಗೇ ಆಗುತ್ತೆ. ರಾಡಿಕಲ್ ಮೇಲೆ ಪ್ಲ್ಯೂಮ್ಯೂಲ್ ಬರುತ್ತಾ, ರಾಡಿಕಲ್ ಮೇಲೆ ಕೋಲಿಯೋರೈಸಾ ಬರುತ್ತಾ, ಕೋಲಿಯೋಪ್ಟೈಲ್ ಬರುತ್ತಾ ಅಂತ. ಈ ಕನ್ಫ್ಯೂಷನ್ ಎಲ್ಲರಿಗೂ ಆಗೇ ಆಗುತ್ತೆ. ಎಷ್ಟೇ ಕಾನ್ಫಿಡೆಂಟ್ ಇರಲಿ, ಎಕ್ಸಾಮ್ ಹಾಲಲ್ಲಿ ಯು ವಿಲ್ ಗೆಟ್ ಅ ಕನ್ಫ್ಯೂಷನ್ ಬರುತ್ತೆ. Now how do you clear the confusion, students? How do you clear the confusion? Now students, understand. I will tell you a small trick. Remember this. Okay, remember this. Here we have coleoptile. Okay, coleoptile. Coleoptile. Then you have coleorhiza. Then we have the coleorhiza. Okay, coleorhiza. Now students, I want everyone to focus. I want everyone to focus on here. In coleoptile we have P. In coleorhiza we have R. P and R. The P you can remember it as plumule. You can remember it as plumule. Then R for radical. R for radical. Okay. So plumule is covered by coleoptile. Radical is covered by coleorhiza. Coleorhiza. So never in your examination you will ever forget what covers what now. Okay. So coleoptile is covered by plumule. Coleorhiza is radical is covered by coleorhiza. So what is the answer? Coleoptile is going to be plumule, plumule and radical. Plumule and radical. Beautiful. Next question. The fruit developed without fertilization is known as. A fruit is developing without fertilization. What do you call that? Do you call that parthenocarpy, amphimixis, apomixis or polyembryony? What is the answer? Quickly tell me the answer in the chat, students. Ellargu 10 seconds kodtini. 10 seconds. Quickly go. The fruit developed without fertilization. A fruit is developing without fertilization. What do you call that? Exactly. Parthenocarpy. Parthenocarpy means developing out of fruits without fertilization. Then what is apomixis? Apomixis is development of seed without fertilization. What is polyembryony? Polyembryony basically means poly means many, many embryo. Polyembryony, many embryo. Okay. Next. Now students, question nodi. Let's see how many of you are able to solve this question. Quickly tell me in the answer in the chat now. This question I have found in a very what do I say? It is not given in your line of NCERT. In the NCERT line kutila, it is there in a diagram actually. It is there in a diagram. Okay. The pistil of papaver is monocarpellary, apocarpus, multicarpellary syncarpus, multicarpellary apocarpus. Let's see how many of you are able to answer this question now. Challenging question. This is a challenging question. Okay, now let's see how many of you are able to answer such a challenging question now. A pistil of papaver is. Let's see how many of you are able to solve this question now. Pistil of papaver. No copying, no copying. Students, this diagram you notidiralla diagram na? Okay. This diagram do legend bunny. When you see the legend of this diagram, can you see here? For your B, this one, multicarpellary syncarpus pistil of papaver. It is given here. Can you see here? Done. Multicarpellary apocarpus. That is the C1. Multicarpellary apocarpus in the case of Michelia. So two examples. Papaver is multicarpellary syncarpus. Multicarpellary means many carpal. Syncarpus means the carpals are fused. Many carpals, the carpals are fused. It is called as multicarpellary syncarpus. What is multicarpellary apocarpus? Multicarpellary apocarpus ಅಂತಂದ್ರೆ many carpels are there, but the carpels are completely free. Carpels are free. So tell me, what is the answer now? Answer is option number C. Done. Next question. The genetic mechanism that prevents self-pollination in plant is? See, there are multiple methods to prevent self-pollination. That is outbreeding devices. Okay, outbreeding devices. In outbreeding devices, which one is the genetic mechanism? Which one is the genetic mechanism? Students, ellaru video na like ಮಾಡಿ first of all, because all of these questions are very beautifully crafted for you to solve. And if you want this PDF or copy of this particular session, it will be available in the Telegram channel. And where is the link to the Telegram channel? The link is given in the description. So go join the Telegram channel to access the PDF copy for free. Okay now? So video like ಮಾಡಿ first of all. The genetic mechanism is going to be your self-incompatibility. ಏನಾಗುತ್ತೆ ಅಂತಂದರೆ ಪ್ಲಾಂಟ್ಗೆ ಅದ್ರ ಮೇಲೆ ಸ್ವಲ್ಪ ಸಾಕಾಗ್ಬಿಟ್ಟಿದೆ. ಪ್ಲಾಂಟ್ ಇಸ್ ಲೈಕ್ ಸಾಕು ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನ್ ಬೇಡ ನನಗೆ. ಐ ಡು ನಾಟ್ ನೀಡ್ ಸೆಲ್ಫ್ ಪಾಲಿನೇಷನ್ ಎನಿಮೋರ್. ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಅಸ್ ಜೆನೆಟಿಕ್ ಇನ್ಕಂಪ್ಯಾಟಿಬಿಲಿಟಿ. ಸರಿನಾ? ದೆನ್ ವಾಟ್ ಈಸ್ ನಾನ್ ಸಿಂಕ್ರನೈಸೇಷನ್ ಆಫ್ ಪೋಲನ್ ಆ್ಯಂಡ್ ಸ್ಟಿಗ್ಮಾ? ಫಾರ್ ಎಕ್ಸಾಂಪಲ್, ನಾನ್ ಸಿಂಕ್ರನಸ್ ಮೀನ್ಸ್ ದ ಪೋಲನ್ ಇಸ್ ರೆಡಿ ಬಟ್ ದ ಸ್ಟಿಗ್ಮಾ ಇಸ್ ನಾಟ್ ರೆಡಿ. ಹುಡುಗ ರೆಡಿ ಆಗಿದ್ದಾನೆ, ಹುಡುಗಿ ರೆಡಿ ಆಗಿಲ್ಲ ಮದುವೆ ಆಗೋದಕ್ಕೆ. ಸರಿನಾ? ಡಿಫ್ರೆಂಟ್ ಪೊಸಿಷನ್ ಆಫ್ ಆಂಥರ್ ಅಂಡ್ ಸ್ಟಿಗ್ಮಾ. ದಟ್ ಈಸ್ ಬಾಯ್ ಇಸ್ ವೆರಿ ಟಾಲ್, ಗರ್ಲ್ ಇಸ್ ವೆರಿ ಶಾರ್ಟ್, ಆರ್ ಗರ್ಲ್ ಇಸ್ ವೆರಿ ಟಾಲ್, ಬಾಯ್ ಇಸ್ ವೆರಿ ಶಾರ್ಟ್. ಡಿಫ್ರೆಂಟ್ ಇನ್ ಹೈಟ್. ಸರಿನಾ? ಪ್ರೊಡಕ್ಷನ್ ಆಫ್ ಯೂನಿಸೆಕ್ಷುವಲ್ ಫ್ಲವರ್ಸ್, ದಟ್ ಇಸ್ ಒನ್ ಕ್ಯಾಟಗರಿ. ಯೂನಿಸೆಕ್ಷುವಲ್ ಫ್ಲವರ್ಸ್ ಮಾಡಬೇಡಿ. ಸರಿನಾ? ಸೊ ಆಲ್ ಆಫ್ ದೀಸ್ ಆರ್ ಔಟ್ ಬ್ರೀಡಿಂಗ್ ಡಿವೈಸಸ್. ದ ಫಸ್ಟ್ ಒನ್ ಇಸ್ ಕಾಲ್ಡ್ ಆಸ್ ಜೆನೆಟಿಕ್ ಮೆಕ್ಯಾನಿಸಮ್. ಸರಿನಾ? ನೆಕ್ಸ್ಟ್ ಒನ್, ಪೆರಿಸ್ಪರ್ಮ್ ಇಸ್ ಫೌಂಡ್ ಇನ್. First of all, what is perisperm? Perisperm is nothing but residual nucellus. A nucellus ಇದೆಯಲ್ಲ, it will become persistent. ಅದು dissolve ಆಗಲ್ಲ, degrade ಆಗಲ್ಲ. So when the nucellus becomes persistent, it gets converted to perisperm. Okay, and the example for perisperm is, the example of perisperm, is it black pepper, wheat, maize, groundnut? Exactly. Akshata, exactly. It's black pepper. It's going to be black pepper. Next question. Long ribbon-like pollen grains are seen in. So students, you artha madkoli. Every single pollination requires a specific character for its pollen grains. For example, a plant is wind pollinated. The pollen grains are lightweight, right? If it is water pollinated, or aquatic plant, it has ribbon-like your pollen grains. And also it will have a mucilage covering. Yak mucus covering go, mucilage covering go, to make sure the pollen grain doesn't become wet. Sarna? So ribbon-like it is found in the aquatic plants. In the case of aquatic plants. Ade ನೀವು insect pollinated ಆದರೆ, if it is insect pollinated, then the flower will be very bright. Nice nectar will be there. Smell, ಒಳ್ಳೆ ಸ್ಮೆಲ್ ಬರುತ್ತೆ. ಜಾಸ್ಮಿದು, ಮಲ್ಲಿಗೆದು. ಸರಿನಾ? So aquatic plants. Next question. Now human reproduction questions again. Next is human reproduction questions. Okay. The first question on the board. The following number of testicular lobules are present in each testis of human. How many testicular lobules are there? So all of you know, in one testis there are 250 compartments. And the 250 compartments are called as what? The 250 compartments are called as testicular lobules. Now inside each testicular lobule, there is 1 to 3 highly coiled structure called as seminiferous tubule. ಹೆಂಗೆ ಅರ್ಥ ಮಾಡ್ಕೋಬೇಕಂತಂದ್ರೆ, there is a large house. In that large house there are 250 rooms. Inside every room there is 1 to 3 window. So large testis, inside the testis there are 250 compartments called as testicular lobules, and inside each lobule there is 1 to 3 highly coiled structures called as seminiferous tubule. So answer really, it said in each testis it is supposed to be 250. Okay, 250 compartments. Next question. A human unpaired male reproductive structure is, see, unpaired structure. Testis is paired, Okay. Seminal vesicle is also paired. Yes. Bulbourethral gland is also paired. Yes. So prostate is unpaired. Prostate is unpaired. Now students, how do I know this? Is it given in NCERT? NCERT ನಿಮ್ಮ ಟೆಕ್ಸ್ಟ್ ಬುಕ್ ಕೊಟ್ಟಿದ್ದಾರಾ ಇದನ್ನ? ಕೊಟ್ಟಿದ್ದಾರೆ ನೋಡಿ ಇಲ್ಲಿ. ಇಲ್ಲಿ ನೋಡಿ ಎಲ್ಲರೂ. Here you can see includes a pair. See, a pair of seminal vesicle, a single prostate, again a paired bulbourethral gland. So bulbourethral gland is paired, seminal vesicle is paired, but prostate is single. Prostate is single, all of you. ನೋಡಿ ಕಾಣಿಸ್ತಿದೆ, ಸಿಂಗಲ್ ಆಗಿದೆ prostate, a single prostate. Sarna? Next question. The nourishing cells in the seminiferous tubule are. So all of you know, inside seminiferous tubule, if you look at a seminiferous tubule, inside there are two type of cells. Inside there are two type of cells. Inside there are two type of cells. One is called as male germ cell, also called as spermatogonia. The other one is called as Sertoli cell. Sertoli cell. ಒಳಗಡೆ ಆಚೆ ಏನಿರುತ್ತೆ, ಆಚೆಗಡೆ we have Leydig cell. Leydig cell. Sertoli cell will do what? Sertoli cell will provide nutrition. Male germ cell will produce sperm. Leydig cell will produce what? Leydig cell will produce testosterone. Testosterone. So questionally, a nourishing cell in the seminiferous tubule. Follicular cell? No. Leydig cell? No. Spermatogonia? No. Sertoli cell? Yes. Next question. After ovulation, on the 14th day, because of LH surge, the structure formed by ruptured follicle is. Nodi, we have the graphene follicle. Graphene follicle rupture ಆದರೆ ಏನು ಸಿಗುತ್ತೆ? Secondary oocyte ಆಚೆ ಬರ್ತಿದೆ. This is the graphene follicle. It is rupturing open and secondary oocyte is released. Once the secondary oocyte is released from the inside, once the secondary oocyte is released, the remaining part gets converted to corpus luteum. Remaining part gets converted to corpus luteum. So corpus luteum. Cerna, corpus luteum. Now I can ask you one more question here. What is the function of corpus luteum? Or what does the corpus luteum produce? What is it producing, students? What is corpus luteum producing? That is a good question again. Now, the first menstrual cycle at puberty is called as. What do you call the first menstrual cycle? Quickly. And students, remember, the menstrual cycle happens only in the case of primates. In the case of primate. Okay. In the case of non-primate, it is estrous cycle. Estrous cycle. So the first menstrual cycle is menarche. Then what is menopause? ಯಾವ್ದು ಹೇಳ್ತೀರಾ? Cutie Pie, Pooja, Mamita, Raksha. What is menopause then? What is menopause? Menopause is the end of menstrual cycle. It happens at 55 to 60 years old. Okay. Then once menstruation, all of you know. Starting is called as menarche. Menarche. Next question. Which of the following cells during gametogenesis is normally diploid? Which of the cell is diploid? See, second polar body, primary polar body, both are in the case of your female things, not here. Now, the question is between spermatid and spermatogonia. What is the answer now? Who can tell me? Who can tell me now? Don't worry, soon we will be starting class 11th also. ನೋಡಿ, ನಿಮ್ಮ ಕ್ಲಾಸ್ 12th ಮಕ್ಕಳಿಗೆ ಸ್ವಲ್ಪ ಜಾಸ್ತಿ ಇಂಪಾರ್ಟೆನ್ಸ್ ಕೊಡಬೇಕಾಗತ್ತೆ ಯಾವತ್ತು. ಕ್ಲಾಸ್ 12th ಬೋರ್ಡ್ ಎಕ್ಸಾಮು, ಸರಿನಾ? ಅವ್ರು ಹಾಗೆ ಮನೆ ಕಳ್ಸೋಕ್ಕಾಗಲ್ಲ, ಸರಿನಾ? So students, spermatid is not the answer. Spermatid is actually haploid. It is a structure which is produced in the spermatogenesis, which later gets converted to sperm by the process called as spermiogenesis. That is, spermatid, which looks something like this, gets converted to gets converted to sperm, that is spermatozoa, also called as the sperm. The process here is called as what? The process here is called as spermiogenesis. Spermiogenesis, not spermatogenesis, beredu. Spermiogenesis. So this also is already haploid because it has already undergone meiosis 1 and meiosis 2. But spermatogonia, students, spermatogonia, male germ cell here. The male germ cell, which is also called as spermatogonia, male germ cell inside seminiferous tubule is diploid. It is diploid. Okay, it is diploid in nature. It is diploid. So spermatogonia is the answer. Okay, is normally diploid. It is normally diploid. Okay. Next question. At the time of implantation aga time alli, sarina, the human embryo is called as. What is the embryo called as? Okay. Now students, remember, before the implantation, there's a process called as cleavage. Now what is this cleavage? In the cleavage, the zygote, which is unicellular in nature, starts to undergo mitosis. 2-cell stage, 1-celled, 2-celled, 4-celled, 16-celled stage, 16-celled stage, right? The 8 to 16 cell stage is also called as your morula. Yes, morula. Now this morula eventually gets converted into blastocyst. Single celled, this is your egg fertilization. 2-celled, 4-celled, this is 8 to 16 cell stage. Then we have blastocyst. If you notice at the blastocyst right here, it has a 2 type of arrangement. All the blastomeres, the cells are called as blastomeres. The blastomeres are arranged in 2 separate layers. The outer layer is called as trophoblast. Can you see the inner part here? In one corner kunti nodi ellado. This is called as inner cell mass. Trophoblast, inner cell mass. This is the structure of a blastocyst, and the same blastocyst implantation. So blastocyst will undergo implantation always. Okay. Next question. Appearance of hair on the head is observed during. When the baby is developing, when is the hair on the head coming? Now students, let me tell you, there is one paragraph I'll show you. From that one paragraph, any of such questions can come to you. For example, when is the first heartbeat? At the end of first trimester, what is happening? At the end of second trimester, what is happening? So all of these questions can come here. I'll show you this is the entire paragraph. Here I want everyone to notice, right? After 1 month of pregnancy, embryo heart is formed. After 1 month, heart is formed, right? By the end of second month pregnancy, the fetus develops limbs and digits. Limbs matlab digits, so fingers baratte. At the end of second month, by the end of 12 weeks, first trimester enagutte, most of the major organ systems are formed. For example, limbs and external genitalia. That is first trimester. Now navella antivi baby kick ಮಾಡುತ್ತೆ ಹೊಟ್ಟೆಯಲ್ಲಿ kick ಮಾಡುತ್ತೆ ಸರಿನಾ. Now what is the kicking? The first movement of the fetus appears, and appearance of hair on the head usually observed during the 5th month. So idu ನಿಮ್ಮ ಆನ್ಸರ್ ಇಲ್ಲಿ ಸರಿನಾ. So first movement hair on the head. By the end of about second trimester, ಇದು important. End of second trimester, the body is covered with fine hair and there is eyelid separation, eyelashes formation. Eyelid separation, eyelids formation by the end of 24 weeks. Sarna? By the end of 9 months, the baby is finally out. That is called as parturition. So in examination, they can ask you a MCQ based on any of the following months. Yen ಆಗ್ತಿದೆ ಈ month ಅಲ್ಲಿ ಅಂತ. Clear? ನೆನಪಿರುತ್ತೆ ಅಷ್ಟು? ನೆನಪಿರುತ್ತೆ ಎಲ್ಲರಿಗೂ? Okay. Next question. This is 5th month. Easy now. Appearance of hair. 5th month. Next question. What does the doctor inject during the induced parturition? ಅಕಸ್ಮಾತ್ ಬೇಬಿ ಆಚೆ ಬರ್ತಾ ಇಲ್ಲ, parturition ಆಗ್ತಾ ಇಲ್ಲ. So what does doctor inject to make sure the contraction is happening? Students, when you were learning about fetal ejection reflex, ಆಗಿದ ತಕ್ಷಣ from the maternal pituitary one hormone is released, no? What is that hormone? From maternal pituitary one hormone is released. That is the oxytocin. Oxytocin. So doctor will inject the oxytocin if the parturition is not happening. Sarna? Nice question. Next question, students. Okay. So we have almost done more than 20 to 25 questions done and dusted already. 20 to 25 questions agogidakle. Now last topic for today, that is your principles of inheritance. Now students, we are trying to cover every single chapter one by one and to make sure you're able to solve the questions and get the concept also. Right? Students, if you're able to solve the questions, let me know in the comment section of this video. Comment ಅಲ್ಲಿ ತಿಳಿಸಿ ನಮಗೆ, is this helpful? What else do you want from us? How can we help you? ಯಾಕಂತ ಅಂತಂದ್ರೆ ನಾವು we are making sure there should be at least a channel which is giving everything for free. We want to make that our goal because that is Vedantu's goal actually. Right? So let us know in the comment section. Tomorrow the biology exam, so was even amazing. Mysore ladre. Sarna. So make sure you watch the other lectures also side by side. Okay. So ellaru comment alli tilisi how it is going to be. Okay. Now in monohybrid cross, the phenotypic ratio in F2 generation will be. Tell me, what is the phenotypic ratio? What is the phenotypic ratio? Quickly, all of you. See, there is something called as monohybrid cross. In monohybrid cross, we have phenotypic ratio. That is 3:1. 3 tall, 1 dwarf. Then we have genotypic. That is 1:2:1. 1:2:1. Okay. Then we have dihybrid. In dihybrid cross, what is the phenotypic ratio, students? What is the phenotypic ratio in the case of dihybrid? 9:3:3:1. 9:3:3:1. Okay. Now what is genotype? They don't ask you this, but I'll tell you a small trick. Okay. The small trick is you just need to multiply. The small trick to find genotype is, now for example, your dihybrid cross is nothing but multiplication of 2 monohybrid. ನೀವು 2 monohybrid cross ತಗೊಂಡು ಕ್ರಾಸ್ ಮಾಡಿ we get dihybrid. For example, ಇದು 3:1 ಅಲ್ಲ, 3:1 into 3:1 you do. 3 3s are 9, 3 1s are 3, 1 3s are 3, 1 into 1 1, 1 into 1 1. Okay, so 3 into 3 9, 1 into 3 1, 3 into 3 9, 3 into 1 3, 1 into 3 3, 1 into 1 is 1. Now similarly, if you do for dihybrid, similarly if you do genotype also, you will get it. That is 1:2:1 into 1:2:1. Tell me, what do we get here? 1 into 1 is 1, 1 into 2 is 2, 1 into 1 is 1, 2 into 1 is 2, 2 into 2 is 4, 2 into 1 is 2, 1 into 1 is 1, 1 into 2 is 2, and 1 into 1 is 1. This is the genotypic ratio. Done. 1 is to 2 is to 1, 2 is to 4 is to 2, 1 is to 2 is to 1. This is the genotypic ratio. Similarly, you need to know what is the phenotypic and genotypic ratio of incomplete dominance. In the case of incomplete dominance, it is 1 is to 2 is to 1 for both phenotypic and genotypic. Same to same. It's equal. In the case of test cross, 1 is to 1 in test cross. ಎಲ್ಲ ಕಲಿಬೇಕು ನೀವೆಲ್ಲ ನೆನಪಿರಬೇಕು ಇದು easy ga 3 is to 1. Next question. A person having genotype IA and IB would show blood grouping AB. This is observed because of, see students, here we know IA is dominant allele, IB is dominant allele. What will happen when there are two dominant alleles present together? Codominance. What happens? Codominance takes place. Codominance takes place that time. Okay, codominance. Hello Tanuja, welcome to the class. Codominance. Next question. The frequency of recombination between gene pair on same chromosome as a measure of distance between genes was explained by. Basically, who told us about map units? Who gave the map units term? Who gave the map units? I'll give you one small clue. The clue is the scientist was actually a student of T.H. Morgan. The scientist which has answered, the scientist is a student of T.H. Morgan. T.H. Morgan is not the answer, obviously. Mendel, Alfred Stewart Went, Sutton and Boveri. Exactly, Alfred Stewart Went. Now students, you tell me, everyone in the chat, Sutton and Boveri, what did they give? They gave some theory, no? ಅವರೊಂದು ಥಿಯರಿ ಕೊಟ್ಟರಲ್ಲ, ಏನೋ ಥಿಯರಿ ಇರ್ತದಲ್ಲ, ಅದು ಕ್ರೋಮೋಸೋಮಲ್ ಥಿಯರಿ ಏನೋ ಇತ್ತಲ್ಲ, ಚಾಟ್ ತಿಳಿಸಿ ಬೇಗ ನಮಗೆ. ವಾಟ್ ಆರ್ ದ ಥಿಯರಿ? ಕ್ರೋಮೋಸೋಮಲ್ ಥಿಯರಿ ಆಫ್ ಇನ್ಹೆರಿಟೆನ್ಸ್. Next question students, next question. Okay, sickle cell anemia is due to replacement of 6th codon GAG by dash in beta globin chain. GAG badlu there is one more codon is coming and sitting. Because of that, valine is being produced. Valine is being produced. GAG was replaced by students. Answer ಬಂದ್ಬಿಡ್ತಾ? Students, ಇಲ್ಲಿ ನೋಡಿ ಎಲ್ಲರೂ, in the GAG, in the 6th beta globin chain, GAG codes for glutamic acid. Okay, now GAG was replaced by GUG, that is coding for valine now. Now because of this one small point mutation, point change, because this point change, the normal red blood cell is getting converted to sickle shaped cell. That is because of GUG, which codes for valine. Okay, next question. The family pedigree of Queen Victoria. The family pedigree, not pedigree biscuit. The family pedigree of Queen Victoria shows a number of descendants for which of genetic disease. ಆ ಫ್ಯಾಮಿಲಿಯಲ್ಲಿ ಒಂದು ಜೆನೆಟಿಕ್ ಡಿಸೀಸ್ ಇದೆ. ಎಲ್ಲ ಫ್ಯಾಮಿಲಿ ಮೆಂಬರ್ಸಲ್ಲೂ ಒಬ್ಬೊಬ್ರಿಗೆ ಬರ್ತಾನೆ ಇರ್ತದು, ಎಸ್ಪೆಷಲಿ ಮೇಲ್ಸಿಗೆ ಬರುತ್ತೆ ಜಾಸ್ತಿ. ಯಾವುದಪ್ಪ ಅದು? ಇಟ್ಸ್ ಆಲ್ಸೋ ಕಾಲ್ಡ್ ಎಸ್ ಎ ಬ್ಲೀಡರ್ಸ್ ಡಿಸೀಸ್. ಒಂದು ಕೈ ಮೇಲೆ ಒಂದು ಕುಯ್ದುಬಿಟ್ರೆ, ಒಂದು ಕಟ್ಟಾಗ್ಬಿಟ್ರೆ ಸಾಕು, ಫುಲ್ ಬ್ಲೀಡಿಂಗ್. ದ ಬ್ಲೀಡಿಂಗ್ ವಿಲ್ ನಾಟ್ ಸ್ಟಾಪ್ ಓನ್ಲಿ. ದಟ್ ಈಸ್ ಯುವರ್ ಹೀಮೋಫೀಲಿಯಾ. Nice. Hemophilia. Next question students. Students, ಎಲ್ಲರೂ ಚಾಟಲ್ಲಿ ಆನ್ಸರ್ನ ಲೈವ್ ಕೊಡ್ತಾ ಇರಬೇಕು, ಆವಾಗಲೇ ಮಜಾ ಬರೋದು. ಸಿ ವಿ ಕೂಡ ಮೇಡ್ ದಿಸ್ ಸೆಷನ್ ಆಸ್ ರೆಕಾರ್ಡೆಡ್. ನಾವಿನ್ನ ರೆಕಾರ್ಡ್ ಮಾಡಿಬಿಟ್ಟು ನಿಮ್ಗೆ ಕಳಿಸ್ಬೋದಾಗಿತ್ತು, ನೀವು ಖುಷಿಯಾಗಿ ನೋಡ್ಬೋದಾಗಿತ್ತು ರೆಕಾರ್ಡೆಡು. ದ ರೀಸನ್ ವಿ ಆರ್ ಡೂಯಿಂಗ್ ಇಟ್ ಲೈವ್ ಸೊ ದ್ಯಾಟ್ ಯು ಕ್ಯಾನ್ ಪಾರ್ಟಿಸಿಪೇಟ್ ಇನ್ ಅ ಕ್ವಿಸ್ ಫಾರ್ಮ್ಯಾಟ್. ಯು ಕ್ಯಾನ್ ಪಾರ್ಟಿಸಿಪೇಟ್ ಇನ್ ಡೈರೆಕ್ಟ್ ಆನ್ಸರಿಂಗ್ ಕಾಂಟೆಕ್ಸ್ಟ್, ರೈಟ್? ಕಾಂಟೆಸ್ಟ್ ನಡಿಬೋದು. right? So loss of X chromosome from normal diploid set leads to basically hypoploidy. Okay, does it happen in Down syndrome, Turner syndrome, Klinefelter syndrome, sickle cell anemia, in which one chromosome is missing? I'll give you a small clue. Here we get XO. The female is XO. In the female, one X chromosome is missing. X chromosome is missing. X chromosome is missing. X chromosome is missing here. That is happening in the case of Turner syndrome. Very good. It is happening in the case of Turner syndrome. That is XO. Now, what about in the case of Down syndrome, students? What exactly happens in the case of Down syndrome? Is there addition or deletion? Tell me. Is there addition or deletion in the case of Down syndrome? In the case of Down syndrome, there is a addition of 21st chromosome. Yes. Trisomy of 21. Trisomy 21. Trisomy of 21. Addition. Now, what is happening in Klinefelter syndrome, students? In the case of Klinefelter syndrome, is it again a addition or a deletion? In the case of Klinefelter syndrome, is it not X, X, Y, an extra X chromosome? In the case of Klinefelter syndrome, it causes gynecomastia in the case of males. Yes, XXY. This is your homework question. That's it, students. We have done more than almost 25 plus questions, 27 questions ಮಾಡಿದೀವಿ. ಸರಿನಾ? I wanted to keep this session short. So based on your requirements, we will see if you want to have more sessions like this before your biology exam. So only if you let us know in the comment section. This is your homework question. This is your homework question. Haplodiploidy, sex determination is seen in. Students, I want the homework question in the comments, not here. In the comments. Okay. Homework question in the comments, everyone. So I wanted to keep this session very short because I know multiple students have multiple exams going on. So let me know in the comment section if this session was useful to you, if you were able to follow the questions, if you're able to understand the answers and then solve the questions. If yes, students, quickly smash the like button, subscribe to the channel if you're new, and my name is Baswaraj sir. So if you do not know that, and I will see you in the next session. Keep commenting, keep showing love, keep showing support, and the more support you show, the more effort which we put, right? It is a reciprocal relationship, right? So it's a direct reciprocal. You show more support, we put more effort. Okay, so thank you so much, students, and if we have exam later on, we will do one more session for you. Okay, we will do one more session. So let us know in the comment section if this session was useful. If it is useful, then we will do a part 2. Okay, so take care, students. Bye bye. Have a great day ahead, all of you. Take care and keep studying. Bye bye.